I'm Vicky Stone. I'd like to thank the organisers for inviting me to speak in this meeting this week. I'm going to talk about the grouping and read across of nanomaterials and how this can be used to streamline hazard testing. I'm going to focus on the outputs of two projects, Gracious and Sunshine. So nanomaterials are very diverse in their physical and chemical properties. They come in a range of sizes, shapes and chemistries, all of which can influence their hazard and also their fate and behavior in the environment. And they're also used for a wide range of different applications. Therefore, the potential for exposure for humans in an occupational or consumer setting are quite significant, as well as their release and potential to the environment. So we need to understand their risks, but to do risk assessment, we usually consider substances on a case-by-case basis. This is very expensive, it's time-consuming, and it uses large numbers of animals. And so because of the large number of substances that are available that require this information, the European regulation, REACH, allows you to group together substances which are sufficiently similar to each other in terms of their physicochemical characteristics, their toxicology and their ecotoxicology. So if you then generate a group, you can have a look at the hazard data for your group members, and then that allows you to read across or copy hazard data from one substance to another within the same group if they're sufficiently similar to each other, therefore reducing the amount of toxicology studies required. So within the regulation, there is a lot of talk about doing a similarity assessment, but it doesn't really provide you with clear methods for doing this. And it's often done by expert judgment. But within the Gracious project, we've provided a range of quantitative methods for allowing that similarity assessment to take place. So I've already talked about the guidance document from ECHA that shows you its Appendix R6.1, which describes how grouping can be applied to substances, including nanomaterials. So the Gracious project was set up in 2018, funded by the European Commission, with the following aim. It was to generate a framework that would allow practical application of grouping and the subsequent read across of hazard information between similar nanomaterials. We also use this term nanoforms. This corresponds to different forms of the same nanomaterial. So a nanomaterial might be titanium dioxide, which exists in a number of forms that vary in their size, shape, and coating. So the framework was published in 2020 in the Nano Today journal. This is a simple diagram that as an overview of the framework. It shows how you enter the framework either with a single nanoform for which you'd like to know if a group exists, that this nanoform can be inserted into, or a provisional group of nanoforms that you would like to assess the hazard of and share data in between. You then collect some basic information on these materials that allows you to generate a grouping hypothesis. This is required by the REACH regulation. Once you have a grouping hypothesis, you then need to gather evidence to test your hypothesis, and the framework includes a series of IATAs or integrated approaches to testing and assessment that use tiered testing to generate the data required to either accept your grouping hypothesis, refine it, or reject it and assess the hazard on a case-by-case basis. This framework can be used for regulatory purposes. It can be used to support safe by design approaches to innovation. It can also refine testing strategies and can inform precautionary measures in an occupational or consumer setting. This is a more detailed version of the same framework. So again, you start with a single nanoform or a provisional group. The phys-chem characteristics in the basic information stage include size, shape, and composition. And these values can be either estimated or known values. We also need information on release and exposure scenarios because this informs the route of exposure for humans and the relevant environmental compartments. And we also need information on the purpose of grouping because that allows us to identify the level of certainty and detail of the information used in the remainder of the framework. So for example, In a Safe by Design application, we can use the estimated data from this basic information stage, but for a regulatory application, we would need to use known or measured values. The basic information is then used to generate the grouping hypothesis. We have a range of hypotheses available within the framework that the user can choose from. And we also have a template to allow the user to make their own hypothesis. The hypothesis is then tested by a tailored IATA, an integrated approach to testing and assessment, that allows the user to complete a data matrix that includes all of the data for all of the materials identified, so that the grouping hypothesis can either be accepted, refined, or rejected. The grouping framework aligns with innovation stages, shown here by the stage gate process that goes from early discovery through to the launch of a product, where you have various gates where you make decisions about whether to progress to the next stage or not. So this basic information step allows you to actually cover quite a wide range of stages, from stage 1 scoping through to stage 4 testing and validation, whereas the detailed step allows you to go right through to the launch phase of your product. So there are various different ways in which a hypothesis can be constructed. So we generated a template that guides the user to include the most important information within that hypothesis. So the template includes a life cycle stage, what they are, which is their phys-chem characteristics, where they go, which is their fate and behavior in the environment and their toxicokinetics in the body, and what they do, which are their hazards. And this is within a context of maybe an occupational exposure or an environmental exposure. We generated 17 human hazard hypotheses using this template and 23 for the environment, and all of these have been published. So here are two of the hypotheses as examples. They always include the route of exposure. So here it's inhalation, some information about the phys-chem characteristics. So here we're talking about quickly dissolving materials. And then this is related to the toxicology. So for a quickly dissolving material, we need to consider both the constituent ions or molecules that are released and how they contribute to the toxicity, as well as the nanomaterials. And if they dissolve very quickly, then perhaps we don't need to consider their ability to accumulate within the body. And then for this environmental exposure in the soil, this is for a nanomaterial with a coating, and the hypothesis suggests that you can read across from the uncoated form to the coated form. We also have hypotheses for the gastrointestinal tract and skin, as well as the aquatic environment and sediments. So to test these grouping hypotheses, we have developed these integrated approaches to testing and assessment, which are a series of questions known as decision nodes. If you answer yes to each of these questions, then you can accept your grouping hypothesis. But if you answer no to any of the questions, then the hypothesis is either rejected or refined. To answer these questions or decision nodes, then we have provided some tiered testing strategies with the most relevant standard operating procedures to provide the information required. And quite often we need a threshold value to answer yes or no to each of these questions, but these thresholds can be very difficult to justify scientifically. So as an alternative, we've generated some quantitative methods to assess similarity instead. These decision nodes include questions on whether exposure is likely, on the phys-chem properties, the tissues or environmental compartments that are exposed, and the hazards that are associated with that exposure. Here's an example of one of the IATAs. This is for the oral route of exposure. So you can see the first decision nodes focus on the dissolution rate, ranging from instantaneous dissolution in oral gastrointestinal fluids through to very slow dissolution, measured as a half-life with some cutoff values provided based on information available in the literature. This allows you to identify whether exposure is to the constituent molecules or ions, the particles alone, or a mixture of the two. The next stage would be a consideration of dissolution in the phagolysosomal fluid, which has a lower pH, pH of about 4.5. So then again, you would consider dissolution with different half-lives in order to understand the biopersistence of the particles in the body. This then allows you to assess whether you're considering the toxicity of the particles alone, as in the right-hand side here, or an exposure to a mixture of the particles and the constituent ions or molecules when understanding the toxicity and coming up with the final grouping hypothesis. So this is the framework complete together in one diagram. You can see it includes the dissolution in the oral gastrointestinal fluids, the dissolution inside the cells, and a consideration of the hazards associated with the exposure. For the hazard exposure, we have consideration of cytotoxicity, inflammation and reactive oxygen species, as well as genotoxicity. For each of these decision nodes, we've generated a tiered testing strategy ranging from simple tier 1 methods through to animal models in tier 3. The tier 2 are more complex in vitro models that include multiple cell types, for example, at an air-liquid interface. Quite often, the tier 1 methods are sufficient to provide the data required to allow a yes or no or a similarity assessment for each decision node, but sometimes you need to progress to these higher tiers to provide a higher level of certainty, especially for regulatory applications. This is one of our tiered testing strategies. This one's again for the oral route of exposure. So we have the decision nodes along the top here from the IATA, and then tier 1, tier 2 and tier 3 on the side here. You can see that for every tier we always encourage the users to find existing data, and we suggest specific methods, standardized methods wherever possible for each of the tiers in order to generate the most relevant information. Once the IATA, the user has worked through the IATA, they should be able to populate a data matrix from existing literature, computational methods or laboratory testing to allow them to compare the source material with their target materials in terms of similarity. To assess similarity, we've generated a range of methods to do this in a quantitative manner. These are summarized and explained in a paper by Jeleskova et al., which was published in a special issue of Nano Impact. The remainder of that special issue includes individual papers that focus on specific methods. So just to remind you, within the ECHA guidance, there are 23 mentions of similarity. So this is a really key part of the grouping and read across exercise. So we came up with two different methods for assessing similarity. We have a pairwise assessment and a multidimensional method for assessing similarity. So in this grid on the left-hand side, we have the range of nanomaterials on the horizontal axis, and on the vertical axis it's the same nanomaterials again. So each box allows a pairwise comparison between two materials. So these green boxes with a value of 0 is the material compared with itself. The two rows at the top are benchmark materials, which we know will generate a certain response in this assay, which is a measurement of reactive oxygen species for this particular data set. And then each of these coloured boxes is a comparison between the different nanomaterials, with green indicating a high level of similarity and red indicating a low level of similarity. And we came up with a wide range of algorithms. This is the ordered weighted average in this example for making these comparisons between the data. For the multidimensional method, this allowed us to compare all of the different methods of assessing similarities, all of the different methods for data generation, so cytotoxicity, inflammation, etc., for all of the nanomaterials simultaneously. This method was interesting, generated some interesting results, but wasn't sufficiently robust to support regulatory applications. So in the more recent project, Sunshine, we've been looking at multi-component nanomaterials that include a variety of different substances within an individual material. And so here, the considerations are much more complex than for the nanomaterials considered within the GRACIOS project. So for these multi-component nanomaterials, we need to consider that they have enhanced properties, and there are a wide range of enhanced properties, all of which could influence hazard. But we also need to think about interactions between the components within a multi-component nanomaterial. So substance A might have this hazard here, and substance B might have a similar hazard of a lower level. But when you can put them together, they may just add together so that you have an additive hazard, or there may be a greater than additive hazard known as a synergistic effect, or they may cancel each other out, known as an antagonistic effect. So this means we need to enhance our hypothesis template because it doesn't consider enhanced properties, it doesn't consider interactions or the bioavailability of the different components, and it also, what it didn't do as well is it didn't provide enough information to allow the IATA design or to inform the IATA design. So we now have a new template. This is still in a draft form, but it provides information, background information about the materials. It gathers together the different group members within the template. It explains the purpose of grouping. It also allows us to assess what will be included in the similarity assessment, including the controls. It defines the production, use and life cycle so that we can identify the exposure hotspots. In addition, it includes hazard endpoints related to the mode of action and the adverse outcome pathway. It includes consideration of the enhanced properties and how these impact on where they go and what they do. It includes consideration of the different components within the multi-component nanomaterial and how they might influence where the materials go and where they might separate from each other, and then how they might interact in terms of consideration of hazards. And then all of this information can feed into a generation of decision nodes for the IATA design. At the moment, this template is under discussion in collaboration with two other European projects known as Diagonal and Harmless, and we'll be having a workshop later this month to come up with a consensus about the template. So in summary, we have generated a grouping hypothesis that requires information about phys-chem characterization, fate and toxicokinetics and hazard information, which is in line with the regulatory requirements. And this feeds into a framework that allows these hypotheses to be identified or generated and the evidence identified or generated to test these hypotheses specifically for nanomaterials. We've generated a series of algorithms to allow a quantitative assessment of similarity for nanomaterials so that we can have that evidence to confirm the group members. For multi-component nanomaterials, we've identified that hypotheses need to consider enhanced properties and interactions between the multi-component nanomaterials components. We need to extend the hypothesis template to take these into consideration, and this template also now includes a series of smaller hypotheses or smaller information sections which feed into the generation of the decision nodes for an IATA. For the safe by design purposes, the data matrix doesn't need to be completed for all of the decision nodes. In fact, we can use lower levels of information, which I didn't really talk about today, to support that safe by design process. So I'd like to finish by thanking all of the partners that are involved in both the Sunshine project and the GRACIOS project. And I'd like to thank you for your attention.